Imagine something so small you could fit thousands of them on the head of a pin, yet so resilient they've survived every mass extinction event since they first appeared on Earth over 500 million years ago. Picture a creature that can endure the vacuum of space, withstand radiation levels that would obliterate human cells, and pause its own life for decades, waiting patiently for better days to return. Tonight, we're going to meet one of the planet's most extraordinary survivors, an animal that looks like a tiny bear wandering through forests of moss, but possesses abilities that seem to belong more to science fiction than to science fact. Welcome to Bedtime Biology, where science meets serenity. Each night, we explore the quiet wonders of the natural world, calming your mind and sparking your curiosity. If you like these videos, please consider subscribing to the channel and commenting below if you have any feedback or suggestions for topics of future episodes. The story of how humans first encountered these remarkable beings begins on a crisp autumn day in 1773. Johann August Ephraim Goes, a German pastor and amateur zoologist, was peering through his microscope at a sample of water he'd collected from moss growing on his church roof. What he saw moving through that droplet would puzzle and delight naturalists for centuries to come. There, paddling through the water with 8 stubby legs, was a creature unlike anything documented before. Its plump, segmented body moved with an almost comical slowness, and as Goes watched it lumber along, he was struck by how much it resembled a miniature bear. The resemblance was so uncanny that he gave it an endearing German name on the spot. Kleiner Wasserbär, he called it. Little water bear. Goes's description of these tiny animals sparked immediate interest among the scientific community of 18th century Europe. Letters travelled between natural philosophers, carrying sketches and observations of these peculiar creatures that seemed to exist in a world between the visible and invisible. 3 years after Goes's initial discovery, an Italian biologist named Lazzaro Spallanzani took up the study of these water bears with particular enthusiasm. Spallanzani was already famous for his experiments disproving the theory of spontaneous generation, the idea that life could arise from non-living matter. Now, watching these minuscule animals move with their distinctive plodding gait, he decided they needed a more formal scientific name. Spallanzani chose the Latin word tardigrade, which translates to slow walker or slow stepper. It was a perfect description of their unhurried movement through the microscopic world. If you've ever watched one under a microscope, you understand immediately why this name stuck. They don't dart or zip like many microorganisms. Instead, they lumber along with a ponderous determination, each leg moving deliberately, as if the tardigrade has all the time in the world, which, in a sense, it does. These creatures have mastered the art of patience in ways that put every other life form to shame. The early naturalists who studied tardigrades were working with primitive microscopes by modern standards, yet they managed to document remarkably accurate details about these animals. They noted the segmented body plan, the arrangement of the legs, the structure of the mouth. They observed how tardigrades seemed to prefer living in films of water, clinging to moss and lichen, how they could be found on rooftops and walls, in garden soil and forest floors. What they couldn't have known, watching these creatures through their brass and glass instruments by candlelight, was just how extraordinary their discoveries truly were. they had stumbled upon one of nature's most sophisticated survival machines, an animal that had already been perfecting its remarkable abilities for hundreds of millions of years. In the centuries since Guisa and Spallanzani first described tardigrades, scientists have identified roughly 1,300 different species. That number grows each year as researchers explore new habitats and employ more sophisticated techniques to distinguish between species that can look remarkably similar to the naked eye. Each species has its own particular preferences and adaptations, but all share that distinctive body plan that makes a tardigrade immediately recognizable once you know what to look for. When you first see a tardigrade through a microscope, the bear comparison makes perfect sense. They have a rounded, somewhat barrel-shaped body that gives them a stocky, sturdy appearance. The body is divided into segments, with a well-defined head at one end and 4 distinct body segments following behind. What really drives home the bear resemblance, though, are the 8 legs. Each segment of the body, except the head, bears a pair of short, stubby legs that stick out to the sides. These legs are unjointed, more like fleshy lobes than the articulated limbs of insects or spiders. At the end of each leg, depending on the species, you'll find anywhere from 4 to 8 tiny claws. Those claws are miniature marvels in their own right. Under an electron microscope, they reveal an astonishing variety of shapes and structures. Some species have claws that do indeed look like bear claws, curved and sharp for gripping. Others have evolved elaborate hook shapes, or combinations of claws and adhesive pads, or strange branching structures that look like medieval weapons shrunk down to microscopic scale. These claws aren't just for show. They serve essential purposes in the tardigrade's daily life, providing grip as the animal clambers over moss leaves, helping it anchor itself against currents of water, and in some species, assisting in the capture of prey. The size of tardigrades varies considerably depending on the species, but most fall somewhere between 0.05 millimeters and 0.5 millimeters in length. The very largest species can reach up to 1.2 millimeters, about the size of the period at the end of a printed sentence. This means that while they are microscopic, they're not invisibly small. Under the right lighting conditions, with very good eyesight, a person could potentially spot a tardigrade without magnification. They exist in that fascinating zone of life where they're just barely visible to us, straddling the border between the world we can see and the hidden universe that requires lenses to reveal. The body of a tardigrade is covered in a cuticle, a tough but flexible outer layer that serves some of the same protective functions as an insect's exoskeleton. This cuticle contains proteins and chitin, the same material that makes up the shells of crabs and the wings of beetles. The texture and ornamentation of this cuticle varies tremendously between species. Some tardigrades have smooth, translucent cuticles that allow you to see right through to their internal organs. Others wear armor-plated cuticles decorated with raised bumps, plates, spines, and intricate patterns that look like microscopic artwork. These variations in cuticle structure are so distinctive that scientists use them as one of the primary ways to identify different species. Like all arthropods, tardigrades must shed their cuticle periodically as they grow. This process, called molting, happens multiple times throughout their lives. Some species molt as few as 4 times, while others may molt 12 times or more before reaching their full adult size. During a molt, the tardigrade develops a new, larger cuticle underneath the old one. When the new cuticle is ready, the old one splits open and the tardigrade wriggles out, leaving behind a perfect hollow replica of itself. These shed cuticles are often reused by the tardigrades in surprisingly clever ways, as we'll discover when we look at how they reproduce. The head region of a tardigrade contains some of their most important sensory and feeding structures. Many species have simple eye spots, patches of light-sensitive cells that can distinguish between light and dark but can't form detailed images. These eye spots appear as dark spots on the head and are actually located not on the surface but within the brain itself, peering out through the translucent cuticle. Not all tardigrades have eye spots. Many species that live in permanently dark environments, like deep sediments or underground caves, have lost them entirely over evolutionary time, having no use for vision in their lightless world. Scattered across the head and body, you'll find sensory structures called cirri and clavae. These are delicate, hair-like or club-shaped projections that likely detect chemical signals and perhaps mechanical disturbances in the water around them. Think of them as the tardigrade's version of whiskers, helping the animal sense its environment and locate food even in complete darkness or murky water. The mouth of a tardigrade is unlike anything you'd find on a larger animal. It's a tube-shaped opening at the front of the head, surrounded by lips or lobes that can open and close. Inside this mouth tube are the structures that make tardigrade feeding possible. Two sharp, needle-like rods called stylets can be extended out through the mouth opening. These stylets are made of hardened material and come to extremely fine points. When a tardigrade wants to feed, it positions these stylets against its food source, whether that's a plant cell, an algae filament, a bacterium, or, in the case of predatory species, the body of another microscopic animal. The stylets pierce through cell walls or body walls like tiny hypodermic needles. Once the stylets have made an opening, the tardigrade's muscular pharynx goes to work. This pharynx is a powerful sucking organ, lined with muscles that can create tremendous suction relative to the tardigrade's size. It draws the liquid contents of cells up through the mouth tube, like someone drinking through a straw. Food then passes through a short esophagus into a large midgut, where digestion takes place, and nutrients are absorbed. From there, waste products move into a short hindgut, and are expelled through an anus at the rear of the animal. What tardigrades lack in some ways is as interesting as what they possess. They have no specialized respiratory system, no lungs or gills. They have no heart, no blood vessels, no circulatory system in the conventional sense. Instead, their body cavity, a space called a hemocole, is filled with fluid that bathes all their internal organs directly. Oxygen dissolves into this fluid from the surrounding water through the thin cuticle, and it diffuses from the body cavity fluid directly into the cells that need it. Nutrients absorbed in the gut similarly diffuse through this fluid to reach all parts of the body. This simple system works perfectly well for an animal of their diminutive size, where every cell is close enough to the body surface that elaborate transport systems become unnecessary. The tardigrade nervous system, while simple by vertebrate standards, is remarkably sophisticated for such a tiny animal. They possess a brain, a lobed structure positioned above the pharynx in the head region. From this brain, a nerve cord runs along the underside of the body, with ganglia, clusters of nerve cells, in each body segment. From these ganglia, nerves branch out to the legs and other body parts, coordinating movement and processing sensory information. Recent studies suggest that tardigrade brains might contain fewer than 200 neurons total. For comparison, a honeybee has about 1 million neurons, and a human brain contains roughly 86 billion. Yet, with their mere couple hundred neurons, tardigrades manage all the complex behaviors necessary for survival. They can navigate their environment, locate food, find mates, respond to threats, and make the remarkable decision to enter their death-like survival state when conditions deteriorate. Now, where might you find these remarkable creatures if you went looking for them? The answer is almost anywhere on Earth. Tardigrades have been discovered on every continent, including Antarctica. They've been found at the peaks of the Himalayas, more than 6,000 meters above sea level, and in ocean trenches nearly 5,000 meters below the surface. They live in tropical rainforests and Arctic tundra, in desert soils and mountain streams, in steaming hot springs and permanent ice. Their eggs and dormant forms are so small and so durable that they can be carried vast distances by wind and water, allowing them to colonize virtually every habitat that offers even temporary moisture. That last point is key to understanding tardigrade distribution. While they're among the most resilient animals on Earth, active tardigrades need water. They're truly aquatic animals, even the species we think of as terrestrial. This is because they lack any waterproof barrier on their cuticle. An active tardigrade must be surrounded by at least a thin film of water to prevent uncontrolled desiccation and to allow gas exchange. Without that film of water, they cannot breathe and they cannot move. This dependence on water is why tardigrades are most abundant in habitats that stay moist or that retain water for extended periods. The classic tardigrade habitat is moss. Moss plants grow in overlapping layers, creating a sponge-like structure filled with countless tiny pockets and channels that hold water. When rain falls or dew forms, moss soaks it up and retains it for days or even weeks. Within these moist moss cushions, tardigrades thrive. A single gram of moss might harbor dozens or even hundreds of individual tardigrades, along with the algae, bacteria, rotifers, nematodes, and other microscopic organisms that form their food web. Lichens provide similar habitat. These remarkable composite organisms, formed by symbiotic partnerships between fungi and algae or cyanobacteria, often grow in exposed places like rock faces, tree bark, and roof tiles. Like moss, they have a complex internal structure that can trap and hold moisture. Many tardigrade species specialize in living within lichen communities. Studies in places like the Arctic archipelago of Svalbard have found diverse tardigrade communities within lichen samples, with different species showing preferences for different types of lichens. In freshwater habitats, tardigrades live in the sediments at the bottom of lakes, ponds, and slow-moving streams. They crawl through the spaces between sand grains and organic particles, feeding on the algae and bacteria that coat these surfaces. Some freshwater species never leave the water, remaining active year-round in permanent lakes and rivers. Others live in temporary pools and puddles, places that dry up regularly, forcing them to rely on their survival abilities to wait out the dry periods. Marine tardigrades, numbering about 150 species, occupy a different set of habitats entirely. They live in the spaces between sand grains on beaches, clinging to seaweeds in tidal zones, burrowing into marine sediments. Some have been found at depths exceeding 4,000 meters in the deep ocean. Marine species face different challenges than their terrestrial and freshwater cousins. Instead of drying out, they must cope with changes in salinity and temperature, with the crushing pressure of the deep sea, with periods of freezing in polar waters. Different species have evolved different solutions to these challenges, with some entering dormant states similar to the tun state we'll explore shortly, and others remaining active year-round, but switching between different body forms adapted to summer and winter conditions. The density of tardigrades in favourable habitats can be staggering. Studies have found more than 2 million individuals per square metre in moss samples, and over 300,000 per square metre in soil. These aren't uniformly distributed, of course. Tardigrades cluster where conditions are best, where food is abundant and moisture is reliable. Within a single moss cushion, the population might vary dramatically from one layer to another, from one microhabitat to the next. So what do tardigrades do with their days? What is life like when you're 0.5 millimetre long and living in a forest of moss stems that, from your perspective, tower like redwoods? The answer depends greatly on what species of tardigrade you are. The phylum Tardigrada contains both herbivores and carnivores, grazers and hunters, with many species occupying middle positions on this spectrum as omnivores. The majority of tardigrade species are what scientists call suctorial feeders. They feed by piercing cells and sucking out the contents, using those needle-sharp stylets and that powerful pharynx we discussed earlier. For herbivorous species, the targets are plant cells, algae, and bacteria. A tardigrade will approach a moss leaf or an algal filament, position its mouth against the cell wall, and deploy its stylets. The stylets puncture through the cell wall, creating a tiny hole, and the pharynx begins pumping, drawing out the cytoplasm, the chloroplasts, the dissolved sugars and proteins, everything liquid within that cell. The process looks almost like the tardigrade is drinking from the cell, as if it were using a straw. Different species show distinct feeding preferences. Some feed primarily on the cells of moss plants themselves. Others prefer algae, the single-celled photosynthetic organisms that coat wet surfaces and float in water films. Still, others focus on bacteria, those even tinier microorganisms that live in countless billions in every environment tardigrades inhabit. Some species will feed on a variety of cell types, opportunistically eating whatever they encounter, while others seem to specialize on particular food sources. Then there are the predators. These carnivorous tardigrade species hunt and consume other animals. Their prey includes nematodes, those thread-like roundworms that are ubiquitous in soil and water. They eat rotifers, tiny animals that swim using rings of beating cilia, and that are abundant in the same habitats tardigrades occupy. Some carnivorous tardigrades even prey on other tardigrades, piercing their cuticles and draining their body fluids. There are even reports of cannibalism, larger members of a species consuming smaller individuals of their own kind. Watching a predatory tardigrade hunt, if you're fortunate enough to observe it, is a lesson in patience. These are not fast-moving predators. They rely instead on persistence and opportunity. A tardigrade creeps along with its characteristic slow gait, using its claws to grip the substrate. When it encounters a potential prey item, it must first subdue it. For soft-bodied prey, like rotifers, this might be relatively straightforward. The tardigrade grasps the rotifer with its legs, positions its stylets, and begins feeding. For tougher prey, like nematodes with their resilient cuticles, the process takes longer. The tardigrade must find a vulnerable spot, pierce through, and maintain its grip while the prey struggles. One of the most studied carnivorous species is Milnesium tardigradum, a relatively large and robust tardigrade found in moss and lichen worldwide. Scientists have observed these animals feeding on a variety of prey in laboratory settings, and they've documented some fascinating behavior. When presented with multiple prey items, Milnesium shows clear preferences, choosing certain prey over others based on factors we don't yet fully understand. Size matters, certainly. The prey must be small enough for the tardigrade to subdue, but large enough to provide a worthwhile meal. But there seem to be other factors at play too, perhaps relating to how easy the prey is to pierce, how much it struggles, or chemical signals it releases. The buccopharyngeal apparatus, that combination of stylets, muscle, and supporting structures in the tardigrade's head, is incredibly important for classification and identification of species. Different species have evolved distinctive variations in the size, shape, and structure of this feeding apparatus, and these variations correspond to their different diets. Carnivorous species tend to have larger, more robust structures capable of piercing tough prey cuticles. Herbivorous species might have more delicate stylets suited for penetrating plant cell walls. By carefully examining the buccopharyngeal apparatus under high magnification, scientists can often identify a tardigrade to species level, even when other features look very similar. Movement in tardigrades is another aspect of their biology that seems charmingly simple at first glance, but reveals surprising complexity on closer examination. Those 8 stubby legs work in a coordinated pattern that resembles the gait of much larger animals. Typically, tardigrades use an alternating tetrapod gait, the same basic pattern used by salamanders and many other 4-legged vertebrates. In this gait, the tardigrade moves diagonal pairs of legs together. The front, left, and rear right legs step forward simultaneously. Then, the front right and rear left, creating a rolling rhythmic motion. The fourth pair of legs, the rear most set, point backward rather than to the sides. These legs serve a different function than the forward 3 pairs. While the front legs provide propulsion and steering, the rear legs act as anchors and brakes. They help the tardigrade grip surfaces, maintain position against flowing water, and perform slow-motion acrobatics as they navigate the complex 3-dimensional terrain of their moss and lichen habitats. Despite their name, slow walkers, tardigrades can actually move with surprising speed when motivated. Relative to their body length, they can cover distances at a respectable pace. But what looks like rapid movement under a microscope translates to barely perceptible progress in human terms. A tardigrade traveling at top speed might cover a few centimeters in an hour. In a single day, even with continuous travel, a tardigrade would be unlikely to move more than a meter. This extreme localization means that for a tardigrade, the immediate microhabitat is the entire world. The moss cushion it inhabits might be the only environment it ever knows. But now we arrive at the feature that makes tardigrades truly extraordinary, the ability that has captured the imagination of scientists and the public alike. We must talk about cryptobiosis, and specifically the state called the toon. This is where tardigrades transition from remarkable to almost miraculous, where their biology enters territory that seems to violate fundamental rules about what life requires and what living things can endure. The word cryptobiosis comes from Greek roots meaning hidden life. It describes a state in which an organism's metabolic activity becomes so low that it's almost undetectable, where the normal processes we associate with being alive, breathing, eating, growing, moving essentially cease. The organism isn't dead. Death is irreversible, but it isn't conventionally alive either. It exists in a kind of suspended animation, waiting for conditions that will allow it to resume active life. Tardigrades enter cryptobiosis in response to environmental stress. When the moss cushion begins to dry out, when the water film evaporates, when the temperature drops or rises beyond tolerable limits, when oxygen becomes scarce, the tardigrade must make a choice. It cannot simply continue as it is. Active life under these conditions means death. So instead, it transforms itself. The most studied and most dramatic form of cryptobiosis is anhydrobiosis, life without water. When a tardigrade enters this state, it forms what scientists call a tun. The word tun originally referred to a large barrel or cask, and it describes the shape the tardigrade assumes in this state. The animal pulls its legs into its body, retracts its head, and curls into a compact, barrel-shaped structure, about 1/3 its normal size. This physical transformation is just the visible part of what's happening. Inside, the tardigrade is systematically shutting down nearly all metabolic activity. First, it must deal with the loss of water. An active tardigrade is more than 85% water by weight. In the tun state, it will lose more than 95% of that water. For almost any other animal, this level of desiccation would be instantly fatal. When cells lose water, their membranes collapse and tear. Proteins denature and clump together. DNA strands break, and essential molecular structures disintegrate. It's biological catastrophe. But tardigrades have evolved an arsenal of protective mechanisms that allow them to survive this seemingly unsurvivable process. The rate of desiccation is critical. If a tardigrade dries out too quickly, even its protective mechanisms can't save it. Laboratory experiments have shown that tardigrades dried rapidly in desiccators perish. But when drying happens slowly, over the course of hours or days, the tardigrade has time to activate its defenses. One of the first protective molecules to appear is trehalose, a sugar. Trehalose has remarkable properties when it comes to protecting biological structures from desiccation. As water leaves the cell, trehalose molecules can partially substitute for water, forming hydrogen bonds with proteins and cell membranes that help maintain their shape and prevent them from denaturing. Many organisms that can survive drying out accumulate very high concentrations of trehalose. The brine shrimp Artemia, whose eggs can survive years of desiccation, can have trehalose make up 15% of their dry weight. Some larval insects reach 20%. Tardigrades produce trehalose, but nowhere near these concentrations. The highest recorded in tardigrades is only about 2% of dry weight. This puzzled scientists for years. How could tardigrades survive with such relatively low trehalose levels when other desiccation-tolerant organisms needed so much more? The answer, it turns out, is that tardigrades have evolved additional protective mechanisms that other organisms lack. One of these mechanisms involves a group of proteins called Cahas proteins, standing for cytoplasmic, abundant, heat-soluble proteins. These proteins are unique to tardigrades. They're intrinsically disordered proteins, meaning they don't fold into stable three-dimensional structures the way most proteins do. Instead, they remain flexible and shapeless. When a tardigrade begins to dry out, Sawer-Holtz's proteins are produced in large quantities. As water leaves the cell, these proteins undergo a remarkable transformation. They change from their disordered, liquid-like state into a gel, then into a glass-like solid. This transformation happens at the molecular level throughout the tardigrade's cells. Think of it like this. As the cell loses water, the Sawer-Holtz's proteins essentially vitrify, turning the interior of the cell into a kind of biological glass. This glass encases and protects all the cell's vital components: the DNA, the other proteins, the membranes, the organelles. Everything is held in place, prevented from moving, prevented from degrading. It's molecular scale suspended animation. When water returns and the tardigrade rehydrates, the glass melts back into a gel and then into liquid. The protected structures are released undamaged, and normal cellular function can resume. Other families of protective proteins have been discovered in tardigrades as well. SAHS proteins, MAHS proteins, and others, each apparently playing specific roles in protecting different cellular structures or processes during desiccation. Some associate with cell membranes, helping maintain their integrity. Others protect specific organelles or genetic material. The tardigrade's DNA faces particular challenges during desiccation. DNA is a long molecule, and when cells dry out, the DNA strands can break into pieces. For most organisms, this level of DNA damage would be lethal, but tardigrades have another unique protein called DSUP for damage suppressor. This protein binds to DNA and shields it from damage during desiccation and from other threats like radiation. We'll return to DSUP when we explore how tardigrades survive radiation, but in the context of the ton state, it's yet another layer of molecular armor protecting the tardigrade's genetic blueprint. There are also changes at the level of gene expression. As the tardigrade enters its tun, certain genes are turned on and others are turned off. Genes involved in normal metabolic processes, in cell division, in protein synthesis for growth, these are shut down. The cellular machinery for copying DNA and making new proteins is mothballed. Meanwhile, genes for stress response proteins, for protective molecules, for DNA repair systems, these are ramped up. The tardigrade is essentially reprogramming itself from an organism configured for active life to an organism configured for survival. And then there's the metabolic slowdown itself. In the ton state, a tardigrade's metabolism drops to less than 1/100% of normal levels. Some estimates put it even lower than that. At such reduced metabolic rates, the energy requirements become almost negligible. The tardigrade isn't eating, isn't moving, isn't growing, isn't reproducing. It's simply existing, maintaining the absolute minimum level of molecular activity necessary to remain poised for revival. This state of cryptobiosis can last an astonishingly long time. Dried tardigrades have been revived after decades of storage. One famous experiment involved moss specimens collected and stored in a museum in the 1920s. More than 100 years later, scientists moistened the moss and observed tardigrades emerging from their tons. Most died within minutes, their century-long survival finally exhausted. But some survived for hours, a testament to the durability of the ton state even across such vast timescales. The longest confirmed survival in a ton state is still being determined, but research suggests that tardigrades could potentially remain viable in this state for many decades, possibly even a century or more under ideal conditions. The limiting factors are probably cumulative molecular damage that occurs even at such low metabolic rates, background radiation slowly degrading their DNA, and the gradual breakdown of protective molecules over time. But for an animal measured in fractions of a millimeter, to survive for decades or a century is the equivalent of a human surviving for tens of thousands of years. It stretches the definition of what we mean by lifespan. When conditions improve and water returns, the revival process begins. A completely desiccated tun can begin showing signs of life within minutes of rehydration. The protective glass-like substances dissolve. Water floods back into cells, plumping them up, restoring their normal shape and volume. Membranes reform their proper structures. Proteins refold. Metabolic processes restart. Within hours, the tardigrade can be walking around, feeding, behaving as if nothing extraordinary has happened. This ability to survive desiccation is why tardigrades can live in temporary habitats that would be unsuitable for most animals. A moss cushion on a sun-baked rock might be wet for only a few weeks each year. The rest of the time, it's bone dry. For most animals, this would be an impossible place to live. But for tardigrades, it's routine. They simply time their active periods to match the wet periods and spend the dry months or years in their tuns, waiting patiently for the next rain. But desiccation resistance is only one facet of tardigrade toughness. In the tun state, they also become extraordinarily resistant to temperature extremes. Active tardigrades have temperature tolerances similar to other small animals. They function best in moderate temperatures, typically between roughly 5 and 30 degrees Celsius, depending on the species. But in the tun state, those limitations vanish. Tardigrades in tuns have survived exposure to temperatures as low as -272 degrees Celsius, just 1 degree above absolute zero, the coldest temperature theoretically possible. At this temperature, all molecular motion nearly ceases. Water, if there were any, wouldn't just be frozen. It would be locked in a crystalline solid harder than steel. Yet tardigrades survive. When warmed and rehydrated, they revive. At the other extreme, tardigrades in tuns have survived temperatures exceeding 150 degrees Celsius, hot enough to boil water and cook most organic material. The exposure time matters, of course. Brief exposures can be survived more easily than prolonged heating. But even exposure to 100 degrees Celsius for an hour or more has been survived by some species in the tun state. How do they survive such temperature extremes? The same mechanisms that protect against desiccation provide much of the protection against temperature stress. The vitrification of the cell interior means there's no liquid water to freeze and expand, damaging cell structures. The protective proteins shield against both cold and heat-induced denaturation. And the nearly complete shutdown of metabolism means that the chemical processes that would normally be disrupted by extreme temperatures simply aren't running. Tardigrades have also shown remarkable resistance to high pressure and to vacuum. They've been subjected to pressures exceeding 6,000 atmospheres in laboratory settings, pressures 6 times greater than those in the deepest ocean trenches, and survived. They've been exposed to near perfect vacuum, the kind of conditions found in low Earth orbit, and survived that too. Again, the tun state provides the key. With no air spaces in their bodies to compress or expand, with their molecular structures locked in protective glasses, extreme pressures and the absence of pressure both become survivable. And then there's radiation. This is perhaps the most remarkable aspect of tardigrade hardiness, and the one that has the most intriguing implications for understanding the molecular basis of their resilience. Radiation damages living cells primarily by breaking DNA molecules and by generating reactive oxygen species, highly reactive molecules that can damage proteins, lipids, and genetic material. For most animals, exposure to high doses of ionizing radiation is quickly fatal. The radiation dose that would kill 50% of humans is around 4 to 5 gray. A gray is a unit measuring absorbed radiation dose. Most tardigrades can survive doses of 5,000 to 6,000 gray, more than 1,000 times the lethal human dose. How do they do it? Part of the answer lies in that DSUP protein we mentioned earlier. Damage suppressor protein binds to DNA and somehow shields it from radiation-induced breaks. Researchers have transferred the DLUP gene into human cells growing in laboratory cultures and found that those cells became significantly more resistant to radiation damage. The exact mechanism is still being worked out, but it appears that DSUP may help stabilize DNA structure and perhaps enhance DNA repair processes. But DSUP is only part of the story. Recent research has identified other genes and proteins involved in radiation resistance. Some tardigrade species produce pigments called betalains when exposed to radiation. These pigments act as antioxidants, neutralizing the reactive oxygen species generated by radiation before they can cause cellular damage. When researchers treated human cells with these tardigrade-derived betalains, the cells showed enhanced survival after radiation exposure. Tardigrades also have robust DNA repair mechanisms. All organisms have systems for repairing damaged DNA. It's essential because DNA damage occurs constantly from various sources, not just radiation. But tardigrades seem to have particularly efficient repair systems. They can rapidly detect and mend breaks in their DNA strands, stitching the molecules back together before the damage becomes lethal. There's also evidence that some tardigrade proteins might help reorganize the DNA during stress, packing it more densely and protecting it from damage. The interplay of all these mechanisms, shielding proteins, antioxidant pigments, rapid repair systems, and possibly others we haven't discovered yet, gives tardigrades their extraordinary radiation resistance. This leads us naturally to what might be the most famous chapter in tardigrade biology: their journey to space. In September 2007, a Russian spacecraft called Photon M3 launched into low Earth orbit carrying a very unusual payload. Packed into special containers aboard the spacecraft were several thousand tardigrades, representing 2 species, Rictursius coronifer and Milnesium tardigradum. This mission had a simple but profound goal: to see if tardigrades could survive direct exposure to space. The tardigrades had been placed in their tun state before launch. Once the spacecraft reached orbit, about 270 kilometers above Earth's surface, automated systems opened the containers, exposing the tardigrades to the vacuum of space. For 10 days, they floated in this most hostile of environments. There was no air, no pressure, no water. Temperatures fluctuated wildly as the spacecraft orbited between sunlight and shadow. They were bombarded with cosmic rays, high-energy particles from distant stars and galaxies. They were exposed to unfiltered solar radiation, including intense ultraviolet light that would be deadly to most life forms. The experiment included several different treatment groups. Some tardigrades were shielded from ultraviolet radiation, but exposed to vacuum and cosmic rays. Others received varying levels of UV exposure. When the spacecraft returned to Earth and the tardigrades were rehydrated, the results were both encouraging and sobering. Nearly all of the tardigrades that had been exposed to vacuum and cosmic rays alone survived and went on to reproduce normally. The vacuum of space, it turned out, was no barrier to tardigrade survival. The UV radiation proved more challenging. Tardigrades exposed to moderate levels of UV showed reduced survival, and those exposed to the full spectrum of solar UV radiation, more than 1,000 times more intense than UV exposure on Earth's surface, suffered heavy losses, but not total losses. Even under these most extreme conditions, some individuals survived, reanimated, and produced viable offspring. This experiment, called TARDIS for tardigrades in space, made headlines worldwide. Tardigrades became the first animals confirmed to survive direct exposure to space. Previous experiments had shown that bacterial spores and lichens could survive space exposure, but no animal had achieved this before. The implications were profound, not just for understanding tardigrade biology, but for astrobiology, the study of life in the universe. If tardigrades could survive in space, could they potentially survive transport between planets? Could they ride on asteroids or meteorites, dormant in their ton state, and potentially carry life from one world to another? This idea, called panspermia, has been debated for over a century. The tardigrade space experiments didn't prove panspermia was possible. There are still major obstacles, including the heat and shock of atmospheric entry. But they did show that at least some Earth organisms could survive the vacuum and radiation of space for extended periods. Following the Photon M3 mission, tardigrades have been sent to space several more times. In 2011, they flew aboard the International Space Station as part of an experiment examining how multiple generations would fare in the microgravity environment. More recently, in 2021, tardigrades were sent to the ISS again to study how their genes respond to the combined stresses of microgravity and radiation over multiple life cycles. One tardigrade space story didn't have such a happy ending. In April 2019, an Israeli lunar lander called Bereshit attempted to land on the moon. Aboard the lander was a capsule containing thousands of dehydrated tardigrades. The landing didn't go as planned. Bereshit crashed into the lunar surface at high speed. The crash sparked immediate speculation about whether the tardigrades could have survived and whether the moon might now be contaminated with Earth life. The reality is almost certainly less dramatic. While tardigrades can survive tremendous pressures, the shock of the impact probably exceeded even their impressive limits. Research suggests that impact pressures above about 1.14 gigapascals would be lethal even to tardigrades in tunes. The Bereshit crash likely generated pressures far higher than this. Even if some tardigrades did survive the impact, the moon's surface is an extraordinarily hostile environment. The lack of liquid water means there's no way for the tardigrades to rehydrate and revive. They would remain as dormant tons indefinitely, and even in that state, the moon's intense radiation, extreme temperature swings, and micrometeorite bombardment would gradually destroy them. Still, the incident raised important questions about planetary protection. As we send more missions into space and potentially establish bases on the Moon and Mars, how do we prevent contamination of these pristine environments with Earth life? Tardigrades, with their extreme resilience and microscopic size, represent exactly the kind of organism that that could accidentally hitch a ride on a spacecraft? These are questions space agencies continue to grapple with as spaceflight becomes more common. But let's return from the cold expanse of space to the warm, wet world where tardigrades evolved and where they carry out another essential task: reproduction. Like many aspects of their biology, tardigrade reproduction shows remarkable diversity across species. Sexual reproduction is common, with distinct male and female individuals in most species. But parthenogenesis, reproduction from unfertilized eggs, is also widespread. Some species reproduce exclusively through parthenogenesis, having no males at all. Others can switch between sexual and asexual reproduction, depending on environmental conditions. In species with sexual reproduction, the process can be fascinatingly complex. Tardigrades have a single gonad, either an ovary in females or a testis in males, positioned above the digestive tract. When it's time to breed, which for most species means when conditions are favorable and food is abundant, the sexes must find each other. Given their slow movement and limited ability to detect things at a distance, mate finding is probably largely a matter of chance encounter. Some species show courtship behavior. Males and females engage in what looks like a gentle dance, stroking each other with sensory structures, possibly assessing each other's suitability as mates. In other species, the process appears more perfunctory, with mating occurring almost immediately upon encounter. The mechanics of mating vary widely. In some species, the male deposits sperm into the female's genital opening, and fertilization occurs internally. In others, the male somehow manages to inject sperm directly through the female's cuticle into her body cavity, where the sperm find their way to the unfertilized eggs. This might sound bizarre, but it solves a practical problem. Tardigrades have no specialized copulatory organs, no structures equivalent to those found in insects or other arthropods. Piercing the cuticle, using perhaps the same stylids used for feeding, provides a way to transfer sperm without such structures. One of the most remarkable mating strategies involves the female's molt. As we discussed earlier, tardigrades shed their cuticle periodically. Some species have evolved to time egg-laying with this molting process. The female develops mature eggs in her ovary. When she's ready to molt, she deposits these eggs inside the old cuticle as she wriggles out of it. The male, if present, then deposits his sperm into the space inside the shed cuticle, fertilizing the eggs. The eggs develop inside this protective casing, essentially using the cast-off cuticle as an egg case. This strategy offers several advantages. The cuticle provides physical protection for the vulnerable eggs. It may help maintain humidity around the eggs in terrestrial environments. And it solves the problem of how to achieve external fertilization when you're a tiny animal living in a complex three-dimensional habitat where water currents might wash away eggs or sperm. Researchers studying this mating strategy in detail have observed that the process can take an hour or more. The male essentially wraps himself around the female's head region, and the pair engage in what appears to be mutual stimulation. The male may ejaculate multiple times over the course of this encounter, ensuring thorough fertilization of the egg clutch. Tardigrade eggs themselves are often strikingly beautiful under high magnification. In species that lay their eggs freely in the environment, the eggs typically have elaborate surface ornamentation. There are eggs covered in spines, in bumps, in ridges arranged in geometric patterns. There are eggs that look like tiny ornate vases, others that resemble miniature sea urchins. These decorations probably serve multiple functions. They may help the eggs attach to substrates, preventing them from being washed away. They may increase surface area for gas exchange. They may even provide some protection against predators or parasites. The eggs hatch after a period that varies with temperature and species, but is typically between 14 and 40 days. If conditions are poor, if the environment is too dry or too cold, the eggs can delay hatching, sometimes for months. When eggs that have been in a desiccated state are rehydrated, they may take up to 90 days to hatch. The embryo developing inside the egg goes through a series of cell divisions and transformations, gradually forming the basic tardigrade body plan. When development is complete, the juvenile tardigrade uses its stylets to cut through the eggshell, emerging into the world as a miniature version of the adult. Juvenile tardigrades look much like adults, but are smaller and may lack some adult features, like the full complement of claws or certain sensory structures. They immediately begin feeding and growing. Because tardigrades are eutelic, meaning they have a fixed number of cells, growth doesn't happen through cell division. Instead, the existing cells simply grow larger. The tardigrade molts periodically, shedding its cuticle and revealing a new, larger one underneath. With each molt, it adds more claws, develops more sensory structures, and matures sexually. The time from hatching to sexual maturity depends on food availability and environmental conditions, but can be as short as 12 days in some species. Others take weeks or months to mature. Once mature, tardigrades may reproduce multiple times over their lifespan. Females can lay several clutches of eggs, usually between 6 and 30 eggs per clutch. In favorable conditions, population growth can be rapid, with multiple generations cycling through in a single season. The lifespan of an active tardigrade is generally short by our standards. Most species live 3 to 6 months in their active state, though some can survive up to 2 years. But here's where tardigrade biology becomes truly remarkable. Time spent in the tun state doesn't count toward their lifespan. A tardigrade that spends half its time in cryptobiosis can potentially live for many years of calendar time while experiencing only a few months of active life. There's a hypothesis called the Sleeping Beauty hypothesis, named after the fairy tale, which suggests that tardigrades essentially don't age while in cryptobiosis. Research supports this idea. Tardigrades that are repeatedly dried and rehydrated show lifespans comparable to those kept continuously active, but only when you count the active time and exclude the dormant periods. This means that theoretically, a tardigrade could live for decades, or even longer, if it spent most of that time in turns, emerging only occasionally for brief periods of activity. In practice, other factors probably limit their lifespan. Predators eat them, diseases affect them, environmental catastrophes destroy their habitats. But the potential is there for extreme longevity through dormancy. Now we arrive at perhaps the most humbling aspect of tardigrade biology. These tiny creatures aren't newcomers to Earth. They've been here, in essentially their current form, for an almost inconceivable span of time. The fossil record for tardigrades is frustratingly sparse. Their small size and lack of hard parts make fossilization unlikely. But we do have a few precious fossils, all preserved in amber, hardened tree resin that trapped ancient organisms and kept them intact for millions of years. The oldest confirmed tardigrade fossil dates to the Cretaceous period, roughly 80 to 90 million years ago. This fossil, found in Canadian amber and named Beornleggi after a character in Tolkien's fiction, is clearly a tardigrade. Under microscopic examination, it shows the characteristic body plan, the legs, the claws, and remarkably, it looks very much like modern tardigrades. 80 million years of evolution have changed these animals very little, but molecular evidence suggests tardigrades are far, far older than even this ancient fossil. By comparing DNA sequences from different tardigrade species and estimating rates of genetic change, scientists can estimate when different lineages diverged from their common ancestors. These molecular clock analyses suggest tardigrades may have originated as long as 600 million years ago in the late Precambrian era. If this estimate is correct, tardigrades predate the Cambrian explosion, that remarkable period starting about 540 million years ago, when most major animal groups first appeared in the fossil record. They were already ancient when the first fish evolved. They had been around for hundreds of millions of years when plants first colonized land. Dinosaurs came and went, a mere flash in tardigrade history. Mammals radiated and diversified. Humans evolved from ape-like ancestors. Civilizations rose and fell. And through all of this, tardigrades were there, largely unchanged, surviving in their moss cushions and lichen patches, enduring whatever catastrophes the planet threw at them. And catastrophes there were. Earth has experienced 5 major mass extinction events in the past 500 million years. The most famous is the asteroid impact that ended the Cretaceous period and wiped out the non-avian dinosaurs 66 million years ago. But there were earlier, even more devastating extinction events. The Permian Triassic extinction, 252 million years ago, killed an estimated 90% of all species on Earth. It was the closest life on this planet has come to complete annihilation. Tardigrades survived it. They survived all 5 mass extinctions. How? Their cryptobiotic abilities are surely key. When disaster struck, whether from asteroid impact, massive volcanism, sudden climate change, or other calamities, tardigrades could retreat into their tons. As long as their habitats weren't completely sterilized, as long as some moss or lichen or sediment remained to harbor dormant tons, they could wait out the catastrophe, however long it lasted. But there's more to it than just tune formation. Tardigrades have another advantage. Their eggs and tons are tiny and incredibly durable. They can be transported vast distances by wind and water. A single moss fragment blown on the wind can carry dozens of tardigrades to new habitats thousands of kilometers away. This dispersal ability means that even if tardigrades were wiped out in one region, populations elsewhere could eventually recolonize the devastated areas once conditions improved. Some researchers have even calculated that tardigrades are likely to outlive humanity by an enormous margin. Barring an absolutely catastrophic event, like the Sun expanding into a red giant and engulfing Earth, which won't happen for billions of years, tardigrades will probably still be here. Even scenarios that would end human civilization, massive asteroid impacts, supervolcanic eruptions, nearby supernova explosions, probably wouldn't eliminate tardigrades. They're simply too resilient, too widely distributed, too good at waiting things out. There's something both humbling and oddly comforting about this realization. We humans, with our technology and our cities and our sophisticated brains, are a very recent addition to Earth's biosphere. Tardigrades were ancient when we were still gleam in evolution's eye. They've persisted through changes we can barely imagine. They'll almost certainly persist long after we're gone, still plodding through their moss forests, still forming their tons when times get tough, still carrying forward a lineage that stretches back across hundreds of millions of years. What can we learn from tardigrades? Scientists are asking this question with increasing urgency. The molecular mechanisms that allow tardigrades to survive desiccation, radiation, and other extreme stresses have potential applications in human technology and medicine. Imagine if we could stabilize vaccines and other biological products without refrigeration by using tardigrade-inspired protective molecules. Many vaccines require constant cold storage, which makes them difficult to distribute in regions without reliable electricity. If we could incorporate CAX proteins or similar molecules, we might be able to create vaccines that remain stable at room temperature for extended periods. Similar technology could extend the shelf life of agricultural products, protect crops from drought stress, or even help preserve organs for transplantation. The radiation-shielding proteins like DS-UP have obvious applications for protecting astronauts on long-duration space missions, where cosmic radiation poses serious health risks. They might also help protect healthy cells during cancer radiotherapy, allowing higher radiation doses to be targeted at tumors while sparing surrounding tissue. Researchers are also studying tardigrades to understand the fundamental biology of aging. If time spent in cryptobiosis truly doesn't count toward aging, what does that tell us about the aging process? Could we identify factors that slow or pause aging in other organisms? It's early days for this research, but tardigrades offer a unique window into these questions. And there's value in studying tardigrades simply to understand life's diversity and resilience. In an era of rapid environmental change, when species are going extinct at alarming rates, tardigrades remind us that life is both fragile and astonishingly tough. The delicate moss ecosystems that harbour tardigrades are threatened by habitat loss, pollution, and climate change. But the tardigrades themselves, those individual animals, possess survival abilities that put our own species to shame. There's a lesson there about the difference between individual resilience and ecosystem stability, about how the toughest organisms still depend on the habitats that sustain them. Perhaps the most important lesson is simply this: the world is full of wonders. A creature you could fit on the head of a pin has survived since before fish walked on land. It can pause its life for decades. It can survive temperatures that would vaporize most materials. It has been to space and back. And it's living in the moss on your roof, in the lichen on that tree in your yard, in the soil beneath your feet. The extraordinary is everywhere, if we know where to look. As you drift off to sleep tonight, think about those tiny water bears lumbering through their forests of moss stems, each one carrying a history older than most life on Earth. Each one a testament to the quiet persistence of living things. They ask nothing from us. They don't need our help to survive. They were here before us. They'll be here after. But in that small fact lies something worth remembering. Life endures. It adapts. It waits patiently. And sometimes the smallest, slowest creatures are the toughest of all. If you enjoyed this journey into the world of tardigrades, please consider liking this video and subscribing to Bedtime Biology for more explorations of nature's quiet wonders. Share your thoughts in the comments below. What aspect of tardigrade biology surprised you most? What other tiny creatures would you like to learn about? Your engagement helps others discover these stories and keeps this channel growing. Until next time, sleep well and dream of microscopic bears walking among the moss.